Good day, students. I am Venadora Villalon of Palawan National School. Today, I will discuss on incomplete dominance.
The incomplete dominance. Did you know how incomplete dominance occurs? And how to interpret the result of incomplete dominance using a Punnett square? Let's be familiarized with these terms. Genes are the basic physical and functional unit of heredity or a hereditary factors. Genes are the segment of DNA which are the instruction for making a protein. Genes also control our inherited traits. An example of this is when you inherit the genes for making the pigments that create your eye color. Alleles are constructing form of genes. Example shows alleles are always in pair. Homozygous refers to the traits in an organism that are the same. There are two kinds of homozygous. Homozygous dominant, which represent capital letter A, and homozygous recessive, which represent small letter a. Heterozygous refers to traits in an organism that are different. A heterozygous represent capital letter A and small letter a. Let us now differentiate dominant trait to recessive trait. Dominant trait refers to an allele that masks the presence of another allele for the same characteristics. While recessive trait refers to an allele that's hidden in the first billion generation or F1 generation. Here are some common dominant and recessive traits. The person with widow's peak is dominant over a person without it. If the color of your hair is dark, it shows dominant trait, while light color shows recessive traits. Bent little finger is dominant over straight little finger. Let's compare phenotype from genotype. Phenotype is the external appearance of an organism, while genotype is the genetic makeup of an organism. Recessive B represents the genotype of the blue eyes, while dominant B is the genotype of the brown eyes. Blue eyes and brown eyes are the examples of phenotype. And now, hybrid and dihybrid. Hybrid is the result of a cross between parents differ in one or more traits. An example of this when you cross purebred Labrador to purebred Poodle. The offspring will Labradoodles. Dihybrid is the result of a cross between parents that are identically hybrid for two traits. The two traits from this cross are seed shape, which are round and wrinkled. The second is the seed color, which is yellow and green. Brown and yellow are dominant, while wrinkled and green are recessive. Using the Punnett square, the result shows that out of 16 offspring, 9 are round and yellow, and 3 are round and green. 3 of these are wrinkled in yellow, and 1 is wrinkled in green. Have you tried mixing red and white paints? If you have, you should have produced a pink color. Similarly, pink 4 o'clock flowers are produced when red and white flowers of this kind are crossed. This was discovered by other scientists after Gregor Mendel. They found out that some genes are not completely dominant. To further understand the concept and meaning of incomplete dominance, let's watch this video.
What do snapdragons, human height, and speckled chickens have in common? Well, they are easily identified as non-Mendelian traits. By non-Mendelian, we mean that, genetically, they are rule breakers. They don't follow the regular Mendelian rule that having a dominant allele means the dominant trait will show. Remember in our guinea pig video, having a dominant allele represented by a capital H meant that the guinea pig would have hair? Only if there was no dominant present, a genotype of little h, would there be a hairless guinea pig. Well, that's a Mendelian trait. What we're going to cover now is when these traits are non-Mendelian and they don't follow these basic rules. So let's first start by snapdragons. We confess that when we heard this word, we thought they were some really amazing kind of creature. Well, they are amazing, but they're flowers, so I don't know, that's not exactly what we envisioned. Well, in snapdragon genetics, there can be 3 phenotypes: red, white, or something in between, pink. It's called incomplete dominance. In incomplete dominance, the dominant allele is not completely expressed when the recessive allele is around. There isn't really a clear dominant allele. If you cross a red flower, and we're going to write it here big R, and a white flower, little r, you are going to get offspring that are big R, little r. But unlike a Mendelian trait, if this is incomplete dominance, that big R allele is not completely expressed when the little r is around. So big R, little r in this case is pink. If you cross 2 pink flowers, big R, little r, like shown in this Punnett square here, you can get offspring that are red, white, or pink. Incomplete dominance is different from codominance. Codominance, like a co-worker, that prefix co should make you think together. They work together, the alleles, that is. For that reason, we like to use different letters entirely to represent the alleles. There is a codominance involving color in some types of chickens. Take a look at this Punnett square. If you cross a black chicken, represented here by BB, and a white chicken, represented here by WW, all the offspring here are BW. BW chickens are both black and white. They're speckled. See, both traits show up. This is the essence of codominance. And you know, what's more awesome than a speckled chicken? Well, except for those silky bantam chickens. By the way, you will find that many people use different formatting for incomplete dominance and codominance. Some people like to use different letters entirely for incomplete dominance, and some people don't. Some people also prefer to do exponents with codominance, and some people don't. Formatting aside, when you're solving these Punnett squares, since we find this really does vary from classroom to classroom, the real concept is that in incomplete dominance, one allele is not completely dominant over the other. So you see this almost in-between phenotype. In codominance, both alleles are expressed. It's the application that matters here. Height is fascinating. In our immediate family, Pinky is taller than Petunia. Our mom is also taller than Petunia. So how does that happen? There isn't just one height gene. There's a lot of genes that determine your height. What I mean by that is that you don't just have a pair of alleles, like big A, that will code for your height. It's more like someone having a genotype of big A, big B, big C, little c, and maybe more genes to ultimately determine your height. and you inherit one allele for each of the height genes from each parent. All of these genes work together to determine your height. Your skin color is also determined by many genes, just like your height. These are called polygenic traits. Poly means many, so many genes coding for one trait is what polygenic means. By the way, both height and skin color can be influenced by environmental factors as well. Nutrition growing up can affect your height, just as spending a lot of time in the sun can affect your skin color. However, this doesn't change the genetics for this trait. And finally, one more thing that we want to talk about. Epistasis. Epistasis is when one gene really depends on another gene for it to even be expressed. I mean, it really depends on this other gene. So let's pick an animal like, a llama. Let's say this llama has a dominant big B allele, which means its wool will be black. So big B, or big B, little b means that it will have black wool. And let's say that if a llama has a pair of recessive alleles, little b, then it will have brown wool. Now, what if there is another gene that controls whether this pigment will even be expressed in the llama in the first place? In this hypothetical example, a llama could have this other gene coded for by big C, big C, or little c in its genotype. However, if a llama has the genotype little c, it will not allow the other gene for wool color to even be expressed. Since we have two genes here, the gene for wool color and then this gene that controls whether the color is even expressed, this calls for our favorite 16 square dihybrid cross. If you notice in this dihybrid, crossing two heterozygote llamas, any llama with big B and big B will typically give a black llama, and little b will typically give a brown llama. In all cases, unless they inherit a little c. If the llama has a little c in its genotype, then the gene for wool color is not expressed, and the llama is albino. This means that no pigment is expressed at all. Pretty interesting. There are many other non-Mendelian traits. We have another video on multiple alleles and a video on sex-linked traits. These are also non-Mendelian. It's kind of fascinating to see what can happen when you have these rule breakers. One last thing, keep in mind that when problem solving in genetics, you do not want to just assume it's non-Mendelian unless you are provided information or clues in the problem that it might be. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
I hope the concept of incomplete dominance is clear to you now. So remember, the incomplete dominance is when a dominant allele or a form of gene does not completely mask the effects of a recessive allele, and the organism's resulting physical appearance shows a blending of both alleles. Codominance is a form of inheritance wherein the alleles of a gene pair in a heterozygote are fully expressed, or both alleles are expressed equally. Before I end my lesson, I will leave you a quote. We are survival machines, robot vehicles blindly programmed to preserve the selfish molecules known as genes. This is a truth which still fills me with astonishment. By Richard Dawkins.